X-linked severe combined immunodeficiency, or XSCID. Severe combined immunodeficiency syndrome is a disorder caused by a mutation on any of the genes that code for the development and function of T cells, B cells, and natural killer cells. XSCID is the most prevalent form of severe combined immunodeficiency syndrome. It is caused by a mutation in the IL-2 receptor gamma gene, or IL-2RG. This gene is located on the X chromosome, and the mutation results in the absence of both T cells and natural killer cells and causes B cells to be defective. Patients diagnosed with XSCID have decreased immune response, which makes them very susceptible to contracting infections.
So let's talk about the diagnosis and symptoms of SCID. SCID patients detected by newborn screenings have an absence or low levels of T cell receptor excision circles, which indicates serious lymphopenia, meaning one has reduced levels of lymphocytes in the blood. All newborn babies today are screened for SCID as it allows for earlier treatment and a high chance of survival. To confirm the diagnosis of SCID, a doctor will perform a complete blood cell count, which calculates the total number of lymphocytes in the blood, and a manual differential, which counts the percentage of each type of white blood cell. There are more than 4,000 lymphocytes per cubic millimeter in a healthy infant's blood in the first few months of life, of which around 70% are T cells. Since SCID patients don't have T cells, they typically have around 1,500 lymphocytes per cubic millimeter. An excessive number of infections in infants is the most revealing symptom of SCID for those who are not detected by the screening as a newborn. However, these infections are not common ones such as a cold, but are more serious such as pneumonia, meningitis, bloodstream infections, and viral respiratory infections. SCID patients are also at a heightened risk of infections by vaccines that are not harmful to toddlers with healthy immune systems, such as those for measles, chickenpox, and rotavirus. If SCID is not treated within the first 2 years of life, it can be fatal.
The IL-2 receptor gamma chain plays a major role in the growth and maturation of natural killer cells, as well as B cells and T cells. The pink chain represents the gamma chain receptor, which is an integral membrane protein that partners with a ligand-specific receptor, which can be seen in green. These two receptors combine and associate to form a functional receptor that can bind to different cytokines. These cytokines include IL-2, IL-4, IL-7, IL-9, and IL-21. The extracellular portion of the protein binds to the cytokine, and the intracellular part transmits signals to the cell's nucleus. The JAK-STAT pathway is activated upon the binding of the cytokine to the IL-2 receptor. The STAT proteins are phosphorylated and translocated into the nucleus to regulate the expression of the target genes. The mutation in the IL-2RG results in the absence of T cells and NK cells and non-functional B cells. The majority of mutations are detected in exon 3, followed by exon 4 and 5. These mutations include missense, nonsense, insertion, deletion, or splice site mutations, which all lead to a defective gamma chain.
XSCID's effect on the body's innate immune response is mostly seen in the disorder's effect on natural killer cells. Due to the mutation on the IL-2RG gene, natural killer cells are unable to recognize cytokines necessary for their development, such as IL-2 and IL-15. IL-2 is important because it upregulates natural killer cell proliferation. IL-15, when membrane bound to a dendritic cell, is essential for natural killer cells proliferation and survival. Without a natural killer cell being able to recognize either of these two cytokines, natural killer cell precursors cannot develop into mature natural killer cells. Because of this, little to no natural killer cells are found in patients with X-linked SCID. This causes the body to be increasingly vulnerable to viral infections. It's the job of mature natural killer cells to identify and kill virally infected cells in the body.
The signaling abnormalities of immunoglobulins are caused by the cytokines IL-4 and IL-21. Normally, follicular helper T cells will secrete IL-4 and IL-21 cytokines, which are important for activated B cells to proliferate and differentiate. IL-4 is secreted by T cells to influence B cell isotype switching. This cytokine is seen in purple and binds to the gamma chain in the IL-4 receptor, which is located on the B cell. This stimulates B cell isotype switching and antibody secretion of IgE and IgG. IgE antibodies bind to the Fc receptors found on mast cells to trigger allergic reactions through the release of degranulation. Mast cells release chemical mediators that induce reactions such as coughing, sneezing, vomiting, and itchiness to expel the pathogen. IgG antibodies are important for mediation antibody response against viral pathogens or bacterial toxins. These antibodies protect the cell by directly binding to the viral protein or binding to the bacterial toxin, thus preventing it from entering the cell. IL-21 is important in the differentiation and function of plasma and memory B cells. This cytokine acts on B cells to activate and continue antibody production. So, if the gamma chain is mutated, then B cells are unable to proliferate, differentiate, and function properly, leading to the inability to fight off infection.
Depicted here is an overview of T-cell development in SCID patients. Thymocytes are derived from hematopoietic stem cells found in the bone marrow and reach the thymus gland through the bloodstream. During the early stage of T-cell maturation, somatic recombination occurs in order to generate T-cell receptors, which allows for recognition of antigens from various pathogens. However, in the case of SCID patients, when defects occur during VDJ recombination, naive T-cells are not properly developed by the thymus, resulting in certain types of SCID conditions. The NK cell receptors are unaffected because they do not undergo VDJ recombination. Let's talk about T-cell immunology for X-linked SCID. The disorder is caused by mutations in the interleukin-2 receptor gamma chain on the X chromosome. This gene encodes for the common gamma chain shared by cytokine receptor complexes. The common gamma chain also plays a role in the functioning of the cell cycle as well as cell proliferation. Mutations in the gene encoding for the alpha chain of the IL-7 receptor disrupt T lymphocyte development without affecting B and NK cells. As a result, IL-7 receptor signaling becomes defective and prevents the development and proliferation of T cells.